I will talk to you today about the tree of life, the diversity of life on Earth, and what we understand of the relationships of living being on Earth, and specifically about the origin of complex life. So classifying life is a very old problem. Obviously, humans have observed that the life on Earth was very diverse, even if we're just talking about the macrobial life, so animals and plants. And there is an entire science that is about classifying organisms. Taxonomy is the study of scientific classification, and in particular the classification of living organisms according to some system that we assume is about their natural relationships. The first to try to come up with such a classification was Aristotle. So he tried to classify all kinds of animals, and he proposed this view that I'm showing here, which was a very hierarchical view that was classifying organisms or creatures in order from the lowest to the highest, and humans being, of course, at the top of this classification. One thing that's very important to keep in mind was that in this classification, this was a fixed classification. There was no concept of evolution, and there was no specific relationship between the different classes of organisms. The organisms had an essence that was fixed and unchanged and unchangeable. So we're doing a really big leap forward, and with the work of Linnaeus. So Linnaeus was a Swedish scientist, and he came up with a new classification, so composed of 3 kingdoms. So this classification contained plants, animals, but also rocks. This was part of the classification of the natural world. And he came up with a system to classify organisms that is basically the one that we still use today, with these 7 ranks in the classification that are displayed here. And if you're familiar with the genus species system, this is still the system that we use today, and this is what he invented. So this is what we call the binomial nomenclature. And so here I just depicted his coat of arms. So he got a coat of arms that had 3 colors on it, and each of them representing animals, plants, and rocks. Of course, so those classifications, well, they were all about what we could observe with the naked eye. And once we discovered microbes, of course, those classifications became different. And we owe this to Anthony van Leeuwenhoek, who is considered to be the father of microbiology. He was a Dutch businessman and scientist, and he invented the first microscope. And so he invented this first microscope and started observing everything he could around him, taking samples from ponds or from everything he could find. And so he observed muscle fibers, bacteria, even if at the time we didn't know what bacteria were, sperm cells, red blood cells, and he made descriptions of those, and he called all of them animalcules, so little animals. And so you might be familiar with this, but really the shift in how we're thinking about classification and how we're thinking about the relationship between different organisms changed dramatically with Charles Darwin. So Darwin was the first time to propose that all existing living organisms are connected, have an evolutionary connection and a relationship, and that all current species have descended from a common ancestor. And that we can represent this through a tree that represents, yes, the connection, the relationships, the family tree, in a way, of all living beings on Earth. So he proposed this concept, but he didn't actually propose a tree with existing, like real species. He just proposed the concept of species being connected to one another. Haeckel was the first one to propose such a tree with like all known species at the time. He was a German scientist, and he proposed this. He was really at the origin of a lot of terms in biology that are still used today. So ecology, phylogeny, so phylogeny being the relationships between species, stem cell, and other words that we still use today. And so he did this mapping of the connections, the relationships between all life forms onto a tree. And so this is what is that you can see here. And in Haeckel's tree, we had 3 main kingdoms. So we had the plants, animals, as expected, and now a 3rd kingdom that he called Protista, or protist. One thing that I want to bring your attention to is that those groups, so those words, we still use them today. They didn't quite mean the same thing at the time. So for example, in plants, we could find fungi. So fungi were considered vegetal life, so plants, but we could also find in this domain of life bacteria that were the photosynthetic bacteria. Whereas in protists, you had basically everything that is, or almost everything that is microbial. So whether they are most of the other bacteria, but also unicellular eukaryotes. So we're going to come back to this. And also sponges, so which are animals, but there are microbial forms of those that were considered protists. Edouard Chaton was a French biologist, and he's the one who proposed a term who made the observation that there are really 2 main classes of cells on Earth. So you have cells that have a nucleus, so the DNA, the DNA of the cell is encompassed in a membrane inside the cell, so inside a nucleus. So this is what we call eukaryotes. So kari means nucleus, so eukaryotes, so with a real nucleus. Whereas all the other cells are called prokaryotes, so they don't have a nucleus. Eukaryotes so have this nucleus, but also are in general much more complex cells, and they have also intracellular other compartments inside the cell that divides the different processes. And this really this became to be recognized as one of the main dichotomy in life on Earth. So after this recognition modified a little bit the way we were thinking about the tree of life. And in this tree, so now we're becoming closer to our time. So in 1969, Whittaker proposed this tree that we still find in some textbooks, where we have plants. We now have fungi that are their own kingdom now. We have animals, but now we're separating. So we have protists. We still have this term, but now we took out the bacteria from protists, and now bacteria are in this phylum that we're calling, or this kingdom that we're calling Monera. So there was this recognition that Monera, so bacteria, prokaryotes, are different types of cells than protista that have a nucleus, that are unicellular eukaryotes with a nucleus. However, you can see on this tree that there is still very much of a hierarchical thinking about life, where bacteria are represented at the bottom, then you have unicellular eukaryotes, and then you have complex life that is represented at the top. Protists, so I've mentioned this term quite a few times now. So again, so for Haeckel, protist was sort of an equivalent of microbial life. It encompassed both eukaryotic microbes and prokaryotic microbes. Today, this is not how we use this term anymore. So here, this is a very schematic representation of what we know of the diversity of eukaryotes, so of all life forms that have cells with a nucleus. And what you can see on this tree is that everything that's not in the purple outline is multicellular. So you have animals at the bottom right, you have fungi not too far from them, and you have plants, land plants at the top. So what you can see is that the very vast majority of eukaryotic life is actually unicellular. And that's what we talk about when we talk about protists. So we're talking about this very vast and diverse group of unicellular eukaryotes. These scientists have proposed different kinds of trees of life. How do we infer those relationships between living organisms? Well, the way we do this is that you need to have traits that are shared between the organisms that you're trying to connect and that you know have been inherited from a common ancestor. And those traits are what we call homologous traits. So they descend from a common ancestor and they diversified with time. And of course, they need to be in all of the organisms that you're trying to compare. So here there's an example that's very obvious. So if you're looking at vertebrates and you're looking at what would be the equivalent of the arm in different species, and you know there's bones that are similar between different species, and they've been inherited from an ancestral vertebrate. And you can look at them and see which ones are more similar to one another, and then assume that those species are more closely related than a species that would have a very different type of arm, for example. Until the 1960s, those traits, those homologous traits shared between species, were difficult to come by in the sense that if you're trying to compare by morphology an animal and a plant, or an animal and a mushroom, that's going to be very difficult or impossible, really. So because of this, because we only had access to the morphology of organisms, what people could do, what scientists could do, was to infer the relationships within clades, so for example, within vertebrates or within specific clades of plants by comparing, I don't know, like the size, the shape of petals. And so we came up with classifications for sub-branches of the tree of life, but not for the tree of life as a whole. And this changed dramatically with the access to molecular data. And so talking about molecular revolution is not an overstatement. So all cells, all living cells that we know, have some universal homologies in how they function, how they encode the genetic information, and how this information is transcribed and translated in the cell into proteins, so that are doing the various functions in the cell. So there's a little scheme here. So all cells encode the information on DNA. This DNA is transcribed into RNA, and this RNA itself is translated in proteins. And so all cells do this with a very similar mechanism. And to do this, for example, the last step, so the translation of the RNA into proteins, all cells will use ribosomes, so those big structures, they're big protein structures that are reading the RNA and translating this into different amino acids, so the building blocks of proteins. And all cells, all bacterial cells, all eukaryotic cells to do this translation, they use the same dictionary. They use the same correspondence between the letters that are on the RNA and what amino acid that corresponds to. And so this ribosome, as I've just mentioned, so the ribosome is a really key structure that is responsible for this translation. It's found in all cells, and it's a big complex that is composed of many proteins, but also of RNA molecules that are embedded in those proteins. And because this is something that you find in every single cell, Carl Woese had the idea of using this, basically this homologous character that you find in every cell, and to try to compare it between different organisms to infer how close they are from one another. So for this, he used one of the RNAs that is found in the ribosome. So this RNA, the ribosomal RNA small subunit, because it's universal, and because it's so important for the cell that those are characters that tend to evolve quite slowly, which is good, because if you're trying to compare organisms that have diverged millions and billions of years ago, you want to have a character that is evolving quite slowly. And because it's such an important protein, such an important structure, it's also something that you find in very high abundance in the cells. So it's actually fairly easy to extract from any kind of cell that you would have and to sequence it. So he used a technique that was developed by Fred Sanger that allowed for the first DNA sequencing. And so how this worked, so this is a pretty ugly image by today's standards, but this was really revolutionary. So what they did here was to extract those RNA molecules that I just talked to you about, that you can find in any kind of cell. And then this molecule, so it's a strain of RNA, so it's letters, it's nucleotides, and you can use an enzyme that will recognize like specific patterns and cut the molecule wherever they find this pattern. And so you can do this, and you will end up with different, like, fragments of your RNA molecule of different sizes. And RNA is charged, like that's a molecule that is charged, and because it's charged, you can migrate it on a gel. We call it a gel. So you use current and electrical current, and you can make those molecules, those fragments, migrate. And because those enzymes recognize specific patterns, and that the RNA is going to be a little bit different in every species, it's going to be similar but not identical, then those fragments are not going to be exactly the same in every species. So you're going to have different fragments, and when you make them migrate, you get this, those gels with all the different fragments that you can observe on this gel. And this is what we call a catalog. And this catalog is or fingerprint is going to be specific to every species that you're looking at. And the idea is basically just to do this for many species, or as many species as you want to compare, and just to look at this fingerprint and see how similar they are between species. And so the more similar they are, the more closely related those species are. And he came up with just a metric to calculate the similarity between those fragments in different species. And he used this to compare a dozen or 15 species at the time, which was already huge. And he wrote this paper that came out in 1977, and that was the first molecular phylogeny, and that allowed us to discover what we still call the third domain of life. So in this paper, again, so they did use the method I've just explained, so to try to compare how similar the ribosomal RNA is between different species, and to calculate, to use this metric of how similar those RNAs are. And if you look at those numbers a little closely, you can see that some species have RNAs that are a lot more similar between themselves than relative to other species. So he found 3 major groups of organisms. And by looking at which those were, he recognized that the first group contained basically what we already knew to be bacteria. Yeah, this was just sort of confirming what we already knew. So bacteria were very different from eukaryotes, and they form a group. He found a second group that was basically corresponding to eukaryotes, so where he found plant, fungi, and slime molds, so unicellular eukaryotes, so protists, that were all forming also a group. However, he realized, or they realized in this paper, that eubacteria, so bacteria, and eukaryotes did not represent the entirety of the diversity of life, and that by doing this metric, they could observe a third group of organisms, which in their species representation corresponded to, at the time, what we called methanogenic bacteria. So bacteria that were living in environments without oxygen and that were generating methane. But from looking at their RNA, we could see that they were as different from bacteria, from more standard bacteria, than they were to eukaryotes. So they were something completely different from a genetic perspective. So they decided to consider them a different kingdom. So this was the third, or different domain of life. So this was the third domain of life. And because they had this sort of unusual metabolism that was based on absence of oxygen generating methane, and this made them think that this was well suited to the type of environment that was supposed to exist on Earth maybe 3 to 4 billion years ago, so something very ancestral. So that's why they call them Archaebacteria, so Archaea meaning old. From now on, I will say Archaea and not Archaebacteria, and I will just come back to this in a second, but basically the idea is that today we know they are not bacteria, they're something completely different, so they are their own domain of life, and they've been sort of renamed, rebranded Archaea alone and not Archaebacteria to avoid any confusion. So for a very long time, and because also this is how they were discovered, Archaea were considered to be extremophile, so liking extreme environments. And it's true that we do find them in very unusual environments, so in environments with very high salt concentrations, very hot environments, like the organisms able to live at the hottest, the highest temperatures are Archaea and not bacteria. They like, or they can like very acidic environments or with very high levels of different metals. So this sort of reshaping of the tree of life into now 3 domains, and with animals and plants actually representing a very small corner of the diversity, was not well accepted at the time. A lot of scientists were upset at the idea that multicellular organisms are actually a minority of life on Earth. So there was a lot of uprisal at the time about this result, but nonetheless, with time, we got to accept that this is really what's representing the diversity of life. So Woos continued to apply this technique, and as we had access to a little more different types of organisms, so he reconstructed, again, using the same method, a tree of life, and he still recovered those 3 domains that are now, so in 1990, he rebranded, so he renamed Archaebacteria into Archaea. So I will talk now a little bit about the environmental sequencing. So as you're probably familiar with, but cellular life is old. We think that cellular life might have originated maybe as early as 4 billion years ago. Eukaryotes, maybe about 2 billion years ago. But multicellular life is actually, like in comparison to all of this, is quite recent, so maybe 800 million years, maybe a little bit more. But nonetheless, it's a recent, cellular life is a recent invention, and which means that the very vast majority of life, whether it's past or present, is microbial. And yeah, so microbial life dominates life on Earth. There's probably no surprise here. It's indispensable for many good things, so nitrogen and carbon fixation. So those are key cycles of life to produce energy that goes from inorganic to organic. That is done by bacteria mostly, and some archaea, of course. Also animal nutrition, so the fact that some termites are able to eat wood, or the rumen of cows, all of their digestion is made possible because they have archaea and bacteria in their gut. Of course, there are bad microbes, so you might be familiar with like food poisoning, of course, Lyme disease, bubonic plague. And there's a lot of unknowns. So we're interested in microbial life because now there's a lot of interest in what we call the microbiome. We know that we're covered in prokaryotes and other also microbial eukaryotes in different parts of our body, and we know that affects us, but we're still not completely sure how. But we know that affects also even potentially our mental health. So there's a lot of interest in microbial life because of this. Microbes are also allowing us to have access to delicious things such as bread, cheese, beer, or to also come up with biofuels. All of this is being made possible because of microbes. And also we have an interest in them because they live in very unusual environments. So as I've mentioned before, maybe some like very sulfurous, very high acidity and very hot environments, or high salt environments. And so scientists are trying to understand how this is possible. And even there's scientists so there's this very nice documentary of this expedition that was meant to sample very poly-extreme environments, so that are both acidic and hot, for example, and to try to determine what are the limits of life. Where do you find life and where do you not find life, and specifically microbial life. But to do this, well, until recently, we were constrained by the fact that to study those microbes, you need to grow them in the lab. But growing even microbes in the lab is not trivial. We don't always know what they need to grow properly. And we estimate that there's only about 1% of existing microbial life that we are able to culture so far. So how do we access the rest of the diversity? How do we study the remaining 99%? Well, this became possible thanks to a technique that was developed by Norm Pace. Norm Pace developed an approach that allows you to take a sample from the environment, a water sample, a soil sample, and to extract the DNA directly from that sample. You don't need to grow any cells, you just extract the DNA. And then you sequence it in a way that I will not develop here, but that allows you to really access, so this RNA, this ribosomal RNA that I've mentioned before, you can now access it, but directly from the environment instead of having to have cultures. And with this ribosomal RNA, we were able to do phylogeny just as I've shown you before, but now on such a much larger diversity of ribosomal RNAs that really showed us how much we were missing. So this is a tree that is from 2013, so it's already starting to be old now, but you can see on this tree that at the time, so although, so this is a tree just of bacteria, and the organisms that the clades for which we had at least one cultured representative at the time are represented in white, and everything else was only known from the environment, so from sequencing directly from the environment. So you can see how much we were missing by only accessing things in the lab. This is a similar tree, more recent, but for Archaea. So similarly here, the dark green triangles are the ones for which we have cultured representatives. So everything else, the very vast majority of archaeal diversity is actually known only from the environment. As we started having access to more and more of this diversity and trying to understand what we call deep relationships, so ancient relationships between lineages, by using those phylogenies of 16S or the ribosomal RNA, we realized that this was actually quite difficult because we're working with one gene, one single gene, and we're trying to resolve all of those relationships. And the problem is that the only way you have to evaluate how close species are or how distant they are is, again, by comparing this gene and see how similar it is between different species, or how different it is. And you can have difficulties retracing those relationships if you are in a situation where species or different lineages diverge very fast. So you've had a very short amount of time during which differences can be imprinted, registered in the molecule that you're looking at. Or if this is combined with divergences that happened a very long time ago, then, well, you might have had differences that allowed you to retrace those relationships, but with time, you can have what we call multiple substitutions. So mutations that happen over and over at the same site, and so eventually the signal, the historical signal, gets erased. A conclusion that trying to reconstruct a universal tree of life just based on this ribosomal RNA was actually impossible, or at least did not allow us to really well resolve the deep relationships between organisms. Again, because we're working with one gene, so it's a limited number of characters. You have multiple substitutions, what we also call saturation. So this leads to what we call poor resolution of those deep relationships. So try to overcome this. We moved from being interested or looking at one gene to try to look at the entire genome of organisms. And this was made possible, again, thanks to technological advances. So at the end of the 90s, we started having access to whole genome sequencing. So the first bacterial genomes were obtained in 1995, first archaeal genome in 1996, the first yeast genome in the same year. And so from there on, having access to more and more entire genomes. And as always, this is pretty exponential. So this is showing you the number of available genomes that were sequenced for different domains of life. So Archaea, bacteria, eukaryotes, here also represented viruses. And this is not to scale because now I've just updated with the number of genomes we have today, and we have over 400,000 genomes, full genomes that are sequenced and available. So how does that help us? Well, now instead of using one gene that we can compare between species, we can try to use many genes. So which genes are we going to use? Well, if you look at any genome, you have more or less, we can classify the genes in this genome into three categories. So you have the core genes that were also called the housekeeping genes. So this is really what is the key functions of a cell. So anything that is related to maintaining, repairing the DNA, doing the expression of this genetic material, so replication, transcription, translation, cell division, obviously. So those are such important genes that you do expect them to be found in all cells and to be conserved across distantly related species. In contrast, you have what we call the flexible shell. So those are genes that will be more to do with how you're interacting with your environment. So what kind of nutrients you take in, how you process those nutrients, how you transport things in the cell, etc., and how you interact with your peers. So those are less well conserved. They're going to be more specific to the type of environment you're in. And so they're more linked, to the the phenotype you have, what kind of metabolism, what kind of nutrient you use and you need. And finally, you have orphan genes. So as their name suggests, so orphan genes are usually genes that we know very little about because they are not found in many species. They're found in a very restricted set of organisms. They tend to be involved in very recent adaptations to an environment. And those are genes that tend to be gained and lost very fast in evolution. And so, of course, if you go back to our question, how do we build a tree of life? Well, which genes are we going to use if we're trying to compare many, very distant organisms? We're going to use the core genes because, again, you do expect them to be universally present. You expect them to be evolving slowly, again, because they are so important that they tend to not be very flexible in terms of changes. And they tend to reflect the history of life. Like those genes are not something that you're just going to exchange with some random organism that is in the environment. This is going to be something that you inherit from your parent cell and her parent cell, etc. So those genes are going to really reflect the relationships between organisms. So how many genes is that? Well, that's actually not so many. It's better than one, but there's probably less than 100 genes that you do expect to find to be conserved across the entirety of life. And again, those genes tend to be involved in what we call those informational processes, such as DNA maintenance and expression. And so, for example, ribosomal proteins. So how do we do this? How do we combine the signal from multiple genes? Well, maybe we'll start by how do we use the signal from a single gene? So we've talked a little bit about this earlier, but now we have access to the actual sequence of genes in an organism. And so you can compare the same gene in many species, again, see how similar they are or how different they are, and try to, we have models to try to understand how they've evolved from an ancestral sequence in a common ancestor, and how this sequence has evolved and diverged in different species as the species evolved. So for example, here we have 4 species. We have the sequence corresponding to the same gene in those 4 species, to a homologous gene. So here, this works like languages, so you can have the word for good morning in different species, and you can see they're similar, but they're not identical. And you can try to infer which letter was inherited from the ancestral word. So for example, the G obviously is the same as everyone, so ancestrally that must have been a G. Now, after that, some species have 2 vowels, so 2 O's, or some only have 1. So here you can infer that there's been either maybe a loss of 1 letter in some species, or there's been a gain of a letter in some species. And so you can do this, and doing this is what we call aligning the sequences. And then Well, sometimes you're not really sure what has happened because maybe there's been a lot of changes and you're not sure which one, what was ancestral, what was not. So you're going to remove those noisy regions. So this is what we call site selection. So you're going to keep just the things for which you're sure that they're inherited from an ancestral character. And then once you have this, then you can look at the similarities between those sequences and then make inferences on the relationships of the species that carry those genes. So here in the end, we have the sequence in species 2 and species 3 that are identical. So we are going to assume that those species are very closely related. In species 1, you only have one difference, whereas in species 4, you have 2 differences with everything else. So we're going to assume that species 4 is the more distant species. And when we do this with 100 genes, well, it's exactly the same. We just combine all the genes with one another. We do the alignments, the site selection, and then you reconstruct a tree in the exact same way. The difference is just that now you have many, many, many more sites to work with. And so this is what people have done. The first tree based on this concatenation, on this like super alignment, was published in 2006. And so they use 31 universal proteins, so ribosomal proteins. And so in this tree, you can see at the time we had access to many more bacterial genomes than anything else. And in this tree, what they could recover was in a way similar to the tree WUS had, except that it had many more species in it. And we have bacteria, archaea, and eukaryotes. This tree, and as in the previous trees, archaea and eukaryotes seem to share are more closely related to one another than they are to bacteria. So now I'm going to focus more specifically on the placement of eukaryotes in the tree of life. Eukaryotic cells, and we've seen this before, they are very different from prokaryotic cells. They're much more complex. They have an intracellular organization that is much more complex than what we find in bacteria and in archaea. They have a nucleus, and they have specificities, like uniqueness of eukaryotic cells, that is, this complex cellular features. But they also share some features with bacteria, and they share some features with archaea. And this is why we often say that eukaryotes have a chimeric nature. For example, one very important feature that they share with bacteria is their membrane. So all cells have a lipid membrane, and the type of lipids that you find in bacteria and in eukaryotes is the same and is very different from the one that we find in archaea. So I won't go into detail here, but they're just completely different types of lipids. One key bacterial feature in eukaryotes are mitochondria. So eukaryotic cells have this compartment inside them that we often refer to as the powerhouse of the cell. This is where most of the energy is being generated. But not just only. There's also many other types of processes very important processes that take place in mitochondria. And this is, all those processes are very similar to what happens in bacteria. And the reason for this is that mitochondria are bacteria, or they're descending from bacteria. So this is what we refer to as the endosymbiont theory. So the fact that mitochondria and also chloroplasts in plants, so what allows for photosynthesis in plants, it's another subcellular compartment, used to be free-living bacteria that were engulfed and sort of tamed and turned into an organelle in eukaryotic cells. This is an old idea, actually. It was first proposed in 1890. It wasn't very well received at the time. People thought that was a crazy idea. But when we start accessing molecular sequences and making phylogenies, we realized, first of all, that mitochondria had DNA inside them. And that if you looked at this DNA and you tried to place it in a phylogenetic tree, this DNA fell in the middle of bacteria, and more specifically in the middle of alpha proteobacteria. So this was the confirmation that mitochondria used to be bacteria. They were involved from an endosymbiosis, like an acquisition of an alpha proteobacterium. And today, well, this still stands even with many, many more genomes that we have access to. People have redone phylogenies many times, and we still find this connection between the DNA that's found in mitochondria and the DNA of alpha proteobacteria. We don't know exactly which lineage, if there was a specific lineage of alpha proteobacteria that gave rise to mitochondria, but nonetheless, we know this is a real connection. So we really have this view of the origin of eukaryotic cells as this host cell that engulfed an alpha proteobacterium that gave mitochondria. Okay, and now if we are looking at the features that are shared, so the other side of the chimeric nature of eukaryotes, so the features that are shared between archaea and eukaryotes. Well, all of those features, or most of those features, tend to have to do with those informational processes. So again, DNA replication, transcription, translation. Many of the components of those informational systems are more similar between archaea and eukaryotes than they are to bacteria. And more than that, actually some of those components only exist in archaea and eukaryotes and don't exist in bacteria. Here it's an example, so an RNA polymerase. So just to show you, so you can see on the left the structure of this complex in bacteria and how it's a lot more, like it's smaller, it's less complex than it is in archaea and eukaryotes. And you can see just from this structure that in archaea and eukaryotes it looks very similar. And on the right side, it's just the list of the different components. And you can see again that the ones that are shared between all three domains of life are a small subset compared to what is shared uniquely between archaea and eukaryotes. So this is something that people have observed very early on when we started knowing that archaea existed. And so this connection between archaea and eukaryotes this evolutionary connection was suspected very early on. Now the question was, well, but which archaea? Are eukaryotes specifically linked to a subgroup of archaea, or are they just sister, what we call like a sister lineage to the entire domain of archaea? For a very long time, until the mid-2000s, there was those two conflicting theories that eukaryotes were either were an independent domain. They were sister to archaea, so this is what you can see on the right. And this is what people refer to as the three-domain tree of life. But there was also competing hypothesis that thought that maybe eukaryotes actually evolved from within archaea, like from a specific lineage of archaea. And so this is what you can see on the left side. And this is what people call the two-domain tree of life, because there were two primary domains, so the bacteria and archaea, and then eukaryotes evolved from inside of archaea. And people were doing phylogenies and were getting conflicting results, and we're not really getting out of this hole because people were always reanalyzing those few universal genes that I've mentioned before. But at the time, this was pre-environmental sequencing, so we had access to very few species. So we didn't have a lot of signal, not a lot of data to work with. And the models, the evolutionary models that we were using were still not very sophisticated, and so we were getting just conflicting results. However, if we're ignoring the phylogenies, but we're just looking at characters, features, there was something that appeared. So here at the top are listed a number of genes that for a long time were thought to be unique to eukaryotes. And if not unique, that are very important for the function of eukaryotic cells. And then when people have looked for homologs of those genes in Archaea, what they found, so this is what you can see with those filled circles. What you can see is that, well, it's not a very clear signal, but you can see that there is a clade, like a very big group of Archaea, so the TAC Archaea, tend to share more characters with eukaryotes than the rest of Archaea. They have homologs for all of, or for many of those eukaryotic signature genes. So more of those are found in TAC Archaea than in Eukaryote. And so because of this, and because we were sometimes getting this in phylogenies also, it really started to be accepted that maybe eukaryotes really do come from inside Archaea, and maybe more specifically sister to TAC Archaea, but this wasn't completely clear. It became a lot clearer thanks to metagenomics, so environmental sequencing, so the access to genomes directly from the environment. And in 2015, so really quite recently, those methods allowed us to have access to a completely new phylum or superphylum of Archaea, so a very large group of Archaea that we did not know so far, and for which we had now access to their entire genome. And when we placed, so the first genome was found in sediments near what we call the, well, what is called the Loki's Castle. And because of this, the very first representative, so this first genome was called Loki Archaeota. After that, we started having more genomes represent, and understanding how diverse this group was. They all have, because the first one was Loki Archaeota, and that's a Norse mythology god, all of them have names after Norse gods that you can see here. But long story short, if we now included those organisms in the phylogenies, now the signal was a lot more clear, and we can see that eukaryotes actually branch not only from inside Archaea, but actually from inside Asgard Archaea. So this was really a revolution. This was a breakthrough when it comes to understanding the tree of life and the origin of eukaryotes. And this was a paper that was published in 2017. And now when we looked at those genomes, and if we're thinking back of those eukaryotic signature genes or signature proteins, and we're updating this presence-absence table, well, what we can see is that those Asgard Archaea have many, many, many more homologs of those eukaryotic signature genes than any other lineage of Archaea, which was like an independent but additional evidence for the connection between those Archaea and eukaryotes. And I'll just finish by saying that, well, all of this was made possible by metagenomics, but we now have the culture of the first representative of those Asgard Archaea, and they show that those Archaea are, from a morphological point of view, they are really quite unusual and complex, which would fit well with the fact that they tend to encode more eukaryotic-like genes, and in particular genes that have to do with cytoskeleton and cell morphology and trafficking. Nonetheless, they are Archaea. They're not some hybrid organism. They are small cells, and they do not seem to have any internal complexity like eukaryotic cells. We're starting to have a clear understanding of the connection between Archaea and eukaryotes. We also know for sure that mitochondria evolved from bacteria. So there is evidence for a fusion event at the origin of eukaryotes. So that involved an archaeal-derived cell and an alpha proteobacterial cell. There's a lot of missing questions, missing answers to the origin of eukaryotes and how the tree of life came to be the way it is now, which is, well, we know there's a connection to Asgards. We still don't know exactly which lineage. Same for the mitochondria. We know that they were related to alpha proteobacteria, but we're not really sure which ones. We don't know yet what was the interaction, what was the driver for this interaction between those two cells that led to this endosymbiosis. And how did this complexification happen? In which order? What drove this complexification of the cellular compartmentation? And was there any role of viruses in the origin of eukaryotes? So those are all pieces of the puzzle that people are currently working on to better understand how eukaryotes came to be and how the tree of life really looks like.